Good evening, ma'am.
Good evening, sir.
Good evening, ma'am.
Sir, you can start now.
Right, ma'am. Good evening, Ajay.
Good evening, sir.
Okay. Shall we start today?
Yes, sir. Good evening,
Okay.
sir.
Okay. Hello.
Yes, sir.
Yeah, good. Good evening, Dhanalakshmi.
Good evening, sir.
Okay. So last class, what we have discussed? Any idea? My dear Ajay, last class you only attended, isn't it? So Dhanalakshmi, you are okay. So today what we are going to discuss about, so transcriptions. So what is meant by transcription, my dear students? Before that transcription, you must know RNA polymerases. RNA polymerases is nothing but one of the enzymes. That enzyme is essential for the synthesis of RNA from DNA molecule. That process is called as what is a transcription. Okay. DNA produce RNA molecule. Without polymerization, without polymerase enzymes, it will not carry out. So before that, what are the RNA polymerases is there, and how it will be involved in the RNA synthesis? What are the factors? That things we are going to discuss. Then only we enter into the transcription process. Okay. So polymerase RNA already you know about the ribose nucleic acid. The RNA synthesized from the DNA molecule with the enzymes. Okay. RNA polymerase, DNA polymerase is also very, very important. The polymerases, enzymes, all are protein molecules. Okay. All are protein molecules. They are having specific structures. Okay. And also a DNA to RNA at the same time, prokaryote as well as eukaryote, they are also different types of polymerase enzymes also present in it. Okay. So they are all comes under the headings known as what is it? Holoenzyme. We will see one by one. Before that, you just see the point. In prokaryotic cell, all three RNA classes, all three type of RNA classes are synthesized by single polymerase. Single polymerase synthesize three RNA classes molecules. Okay. So this is the first point. That means what is the meaning of that one? You know, all the three enzymes also started or produced from the single polymerase enzyme only. Okay. The entire enzyme is called as holozyme. What is by holozyme, my dear students? What is by holozyme? Apozymes. Okay. Already studied in previous sessions. What is by holozyme? Any idea, Dhanalakshmi? Ajay, do you know? Okay. Anyway, I will explain in next slide. So, okay, the entire enzymes, holozymes, consist of a core enzyme. Note it down, my dear students. The whole enzyme consists of core enzymes and sigma factor. These two are combined together and form as a holoenzyme. The entire structure is also called as holozyme. Okay. See that the core enzyme has 5 polypeptide chains. Here, the first one, what is that? Core enzyme, isn't it? The core enzyme has 5 polypeptide chains acting as a catalytic center in the unwinding of the DNA molecules for transcriptions. Unwinding. Okay, it's a mass of cells. It's a group of cells. Sorry, group of genes, chains, like that they may told. Second one is called the sigma factors. The sigma factors, what they did, you know, recognize the promoter region of the DNA for the RNA synthesis. Once again, repeat for you. So before transcription, you must know what are the enzymes involved in this process, particularly RNA polymerase is the enzyme. They produce how many enzymes are there? 3 enzymes is there. It may be vary, depends upon their prokaryote and eukaryote. It consists of 2 parts there. What is there? One is called as core enzyme. Another one is called as sigma factor. The core enzyme, it has a 5 polypeptide chain acting as a catalytic center in the unwinding of the DNA molecules for transcription. Unwinding means, what is that meaning? There is a complete double helical structure present in it. From the double standard helical structure, that can produce which one, you know, RNA molecule can produce from the double standard DNA molecules with the help of the RNA polymerase. So that is sigma factor recognize the promoter region of the promoter region means it is a initiation. Promoter is very, very essential. RNA promoter, isn't it? Here, what they are telling us, sigma factor recognize the promoter region of the DNA. The particular region is essential, isn't it, for the transcription process. That promoter, initial stage, they recognize the sigma factor. This is very, very important. So don't forget, my dear student. Okay. So next one is what is by holoenzyme? Just I gave an introduction, then we will pass on to the next one after that. Holoenzyme is a complete functional enzyme which is catalytically active. Okay. And then holoenzyme consists of an apoenzymes together with its cofactor. Apoenzyme with cofactors. So the holoenzyme is nothing but what is that? The last one, the red ink. See that holoenzyme, that is equal to apoenzyme plus cofactor. So the apoenzyme and the cofactor together is called as holoenzyme. Apoenzyme is nothing but the protein part. Cofactor is the non-protein part. So the apoenzyme is nothing but the protein part. Don't forget, my dear student. Okay. These are the two combined together is called as holoenzymes. See that holoenzyme contain all the subunits required for the functions of an enzyme. Example, DNA polymerases 3, RNA polymerases. So these are the two examples. That's why the RNA polymerases. Now it comes under the heading is known as holoenzyme. Don't forget, my dear students. Once I can repeat for you. RNA polymerase. Okay. Two factor is the core enzyme and sigma factors. Core enzyme, it contains 5 polypeptide chain. They may ask the questions. Sigma factors recognize the promoter region of DNA for the synthesis of RNA. That process is called as transcription. Then what is my holoenzyme? Holoenzyme is nothing but the apoenzyme plus cofactor. Okay. The apoenzyme is nothing but the protein part. Cofactor is the non-protein part. Okay. Both are united together form as a holoenzyme. What are the example? DNA polymerase 3 and RNA polymerase. These are the two example has given as a holoenzyme. Sometimes they may ask the question, given examples, which one of the following is not a holoenzyme? Okay. What is by holoenzyme? Holoenzyme composed of what are the two components? Which one of the following? Like that, they may ask the questions. Okay. Don't forget, my dears. This is very, very important one. See that the core RNA polymerase enzyme, it contains the, what is the 4 components, subunit, 5 subunit is the, see, alpha, alpha, beta, beta dash. The sigma factor is activated on it. As a result, it forms, what is that, RNA polymerase and holoenzyme. Okay. What is the main functions of sigma factor? It catalytic activity. It enhances the rate of the reactions. Okay. As a result, what happens? See that sigma factor is here, alpha factor is here, beta. So 4 components here. So RNA polymerase, holoenzymes. The sigma factor united together form as a RNA polymerase coenzyme. So this one is called as, what is that? Only apoenzyme only, this one. When the sigma factor united, otherwise called as cofactor is united. So as a result of the formation of, what is that? Holoenzyme. Okay. So don't forget, my dear. So this is another part. See that core enzymes. Alpha, alpha, beta, beta. This one is called as, what is that? Okay. So these are the 5 subunits present. When it addition with the sigma factor, it forms as a holoenzymes. This is the non-protein part. This is the protein part. So protein part plus non-protein part gives holoenzymes. Already we saw in the equation format, isn't it? That is very, very important, my dear students. So the rho factor is very, very essential for the identification of the promoter region on DNA. That is very, very important. Okay. Sometimes they may ask the question, sigma factors. Okay. So RNA polymerase structure in details. See here, there are 5 subunits there. Alpha 2, beta and beta dash. And this is called as, what is that? And what is the structure of this one? The symbol. Okay. So this is denoted as what gonna?
Sir, is it omega, sir?
Omega. Omega, this, yeah, correct. Omega. Okay. Alpha and beta, beta dash and omega. Okay. So these are the things. Omega is not like this, I think, my dear students. This is not omega. Omega is it correct? No, omega is some inverted like this. It comes. Okay. Anyhow, I will tell you. So another one, the prokaryotic RNA polymerases is also called as core enzyme. Already know about alpha subunit. The chain initiation and insertion with regulatory proteins. These are all our protein molecules. Already know about. Beta catalytic center. Chain initiation and elongation. That process was done by the beta catalytic center. A beta DAS is a DNA binding. So these are the three is included. Okay. So in the RNA polymerase, they are collectively called as core enzymes. Alpha, beta, beta DAS. Each one, it has a specific activity. Alpha, chain initiation and interaction with regulatory proteins. Beta, catalytic center, chain initiation and elongation processes. And beta DAS, DNA binding process also doing. Okay, see that's all. So this is also another type. This is a different types of polymerase enzymes from prokaryote as well as a eukaryotic nature. The bacterial RNA, given examples of prokaryote, you know about the bacteria is an example. Bacteria, it contains these type of. Okay, what about here? See that 10 eukaryotic, the RNA polymerase 2. RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. Already we saw it, isn't it? Both 3 are involved. Sometimes RNA polymerase 1 and 3 is very, very essential. Suppose if it is not reacted, means the RNA polymerase 2 is involved. So this is the RNA polymerase 2 structures. See the 11 number has given here, isn't it? 1, 2, 3, and 4 and 5 here. 6 and 7 is not the inside it is a collapsed in manner. It's a complex molecules. So up to the 11 subunit present in it in this category. So don't forget, my dear, so RNA polymerase in eukaryotic 11 subunit is there. The bacterial RNA there is a 5 subunit is there. One is called as what is that? Alpha and beta dash, and also and the symbol like this, W like this. This is I will tell you next classes. Okay, what is this? I didn't think. Okay. Go to the next slide. The core RNA polymerase associated with the specific protein factor, the sigma factor. They help the core enzyme recognize and bind to the specific deoxynucleotide sequence, otherwise called as DNA sequence. The sigma factor recognize where it will bind, where the initiation of the transcription takes place. That position, the point will be recognized by the factors called a sigma factor only in the deoxynucleotide sequences only. Okay, this is another one. Second things, RNA polymerase can bind to many regions of the DNA. Not only the only one place, it can bind any places. What is the main purpose of the RNA polymerase? Means what are the sequences, what are the DNA sequences present in it? It completely scans the molecules. RNA polymerase can bind many regions of the DNA, but it scans the DNA sequences at the rate of 103 base per. Okay, 10 to the power of 3 base per second, until it recognizes a certain specific region of the DNA to which it's bind with higher affinity. The bond between the RNA polymerase and also the promoter regions. Okay, it is bind together very highly affinity. Highly affinity, the bond between the two molecules will be very high in nature. That is very, very important things. Okay, see that one. The region is called as promoter, and it is the association of the RNA polymerase. With the promoter that ensure accurate initiation of transcription. Then only the transcription will be what happen. Accurate initiation is done by which one, you know, RNA polymerase. Okay, so suppose but RNA polymerase is not present, in what happen, you know, the transition process occurs inaccurately. Okay, because before that one, what is that? Completely scan the molecule where we will start the transcription process. They can identify which place, then only it will act upon it. As a result, the transcript process initiation taking place. See that one in eukaryotic cells, There are three distinction classes of RNA polymerases. Already told you. How many RNA polymerases molecules present in it? There are three RNA polymerases present in it. RNA polymerase 1, 2, and 3. Each polymerase responsible for the synthesis of different classes of RNA. What is the meaning of different classes? Classes means already tRNA, mRNA, RNA is there. Different classes of RNA they are telling, isn't it? Each one it has a specific responsibility. RNA polymerase 1, 2, and 3. Okay, so don't forget, my dear students. This is very, very important. See that there are three types of RNA polymerase enzymes. See that this one is called as up to that 43, 34 protein is also here. So see here, there are so many things is there. This is RNA polymerase 1, this is RNA polymerase 2, this is RNA polymerase 3. Different structure, different combination, components also present. See that outer sides there is a protein molecule. There are, see that 11. How many types is there? 1, 2, 3, 4, 5, 6. Okay, whatever here. See that one? 1, 2, 3, 4, 5. So different types of RNA. This is the structure. Okay, this will not asked in the diagrammatical aspect questions. They ask the question depends upon their activities, catalytic. Okay, see this one. See the first one. RNA polymerase 1 is responsible for the rRNA, ribosome RNA synthesis. So RNA polymerase 1 is responsible for the ribosome RNA synthesis. Don't forget, my dear student, this is very, very important. Excluding the 5S. The S is denoted as Swedberg unit, otherwise called as sedimentation unit. Okay. 5S ribosome RNA, ribonucleic acid. This is very, very important. Excluding. Okay, this one. So just for note it down. So RNA polymerase 1 is responsible for the rRNA synthesis, excluding this one. So remaining things is also present in it. Don't forget. RNA polymerase 2 synthesize the mRNA. This one is rRNA. This one is called as mRNA. And some of the small nuclear RNAs, as see here, small nuclear RNAs also denoted as snRNA, small letter of snRNAs. Involving in the RNA splicing. What is the meaning of splicing? It may be a fragment nature. It is simply small cut, small nucleotide, small polypeptide chain. Okay, so the RNA polymerase chain 2. And third one is RNA polymerase 3 synthesize tRNAs and single stranded rRNAs and sRNAs. Small nuclear RNAs also synthesizes. This is the T and R. This one is rRNA. This is mRNA. But in this category, tRNA, don't forget, rRNA, mRNA, tRNA. These three are very, very essential for the biosynthesis of the protein molecules. Without three RNA molecules, we cannot be able to synthesize. The nucleus is not synthesize the protein molecules. But at the same time, each and every RNA molecule synthesized by the RNA polymerase enzyme. Don't forget, responsibly. Okay, see that RNA polymerase 1, rRNA. Polymerase 2, mRNA. Polymerase 3, tRNA. Here, you have to add another one, rRNA as well as a single nuclear RNA also. This is more complicated. It has a more active than the previous two. Okay, don't forget, my dear. See this one. The RNA polymerase 1 is responsible for the synthesis of only one type of RNAs. Transcript is called as pre-ribosomal RNA or pre-rRNA, which contain the precursor for the 18S, 5.8S, and 28S and RNAs. Okay, so 18S, 5.8S, and 28S RNAs. These are all are present in where? The bacteria. Okay, see that one? These are all are particular here, exception of 5S RNA. See that one here? It will not come 5S, 5.8S. That's why they have told us excluding. Don't forget, my dear. Go to the next one. So mechanism of RNA. What is the main mechanism RNA polymerase doing? Okay. So this first one, what is meant by polymerase? What are the nature of the polymerase already we studied? Then we will pass on to the next one. What are the types of RNA polymerase? There are 3 types of polymerases there, 1, 2, and 3. What are the responsible of RNA synthesis takes place? Which polymerase synthesis, which type of RNA? That things already we discussed. Next, we will pass on to the mechanism of RNA polymerases. The RNA polymerase produce a transcription unit. Don't forget, my dear. This is very, very important. RNA polymerases produce a transcription unit that extended from the promoter to the termination sequence. Already in our any one of the reaction you can imagine, initiation, elongation, termination is there, isn't it? Likewise, the mechanism of RNA polymerase produce a transcription unit. What is the meaning of unit? That unit, it contains starting point, elongation point, and also termination point. So the unit is nothing but the 3 is important. 3 processes important. See here, the gene is defined is the reference to the start site. Those sequences before the start site are called the upstream sequences, where there is a starting place. That starting place is called as what? Starting site. And starting sites also called as upstream sequences. And those after the start sites are called as downstream sequences. Sometimes they may ask the question, which one is the starting site? That starting site it called as, what is that? Upstream. And those after the start sites are called as, what is that? Downstream. The immediate product is the primary transcript. This is very, very important. Primary transcript, what is meant by primary transcript? When it occurs, like that they may ask the question. I will explain what is meant by primary transcripts. Okay. So before that, we must know that these are the 2 points is very, very important one. Upstream sequences, starting point. After the starting point, what are the things is that? That is downstream sequences. Okay. See that one. And then see the structure, it will be, see means you can easily identify it. So it is not clear in this part. See that? Okay, so I will tell you. Okay, this one. I will tell you, explain it. Okay, see here. Okay, the primary transcripts. See that one. Transcription by RNA polymerase in E. coli. So they have given as an example, bacteria is an example. Particularly all the polymerase enzymes secreted by the E. coli only. Don't forget, my dear students. Okay. For the synthesis of an RNA strand complemented to one of the 2 DNA strands, in the double helicals, the DNA is transiently unwound. About 17 base pair are unwound at any given time. 17 base pair unwound. This is the first step in E. coli transcription. Why they have taken, prokaryotic transmission and also eukaryotic transmission, it will be differ. Okay. In prokaryotic transmission, how many base pair is unwounded? Okay. So 17 base pair is unwounded. RNA polymerase and the bound transcriptions bubbles move from the left to right along the DNA as shown. Facilitations RNA synthesis is transcript. So this is a normal process. How it will be? Already I told you there is a double stranded DNA molecule is there. The RNA polymerase on it. It find out the recognition with the help of the Rho factor and also complete scanning of the molecules, complete scanning of the DNA molecule and find out where the initiation is takes place. That initiation is taking place is called as what is it? Upstream movement. After starting on downstream movement. During that process, how many base pairs are unwounded and any given time means 17 base pairs. Okay. So after that, what happened? The bubbles, it comes out. Okay. I will show you how it will be bubbles or forming. From where to where? Left to right only along the DNA zone. And after that, what happened? Automatically the RNA molecule, single standard RNA molecule synthesized from the DNA molecules. Okay. So with the help of the polymerase enzyme. As the DNA is rewounded, the RNA DNA hybrid is displaced and the RNA strand is extruded. This is the another one. And rewounded means what is the means? there is a small gap in between the DNA strand. RNA started. After that, what happened is this completely, what happened? No, the RNA molecule synthesized in the one stand only, one side only. Okay. After that, what happened? No, it comes out, the DNA once again it united. See, the RNA polymerase, okay, is in close contact with the DNA ahead of the transcription bubbles, as well as with the separate DNA strand and the RNA within the immediately behind the bubbles. I will show you diagram, then only it will be big. See here, this is the structure of the DNA molecule, isn't it? This is the structure of the DNA molecules. So here the active sites is there. So the RNA strand is here. See that this is the RNA strand. The opposites also the RNA strand is there. These two strands. Which one is starting point? You know, here only the starting point. Right to left or left to right. Starting point here. This one is called as what is that? Wounded one. This one is called as unwounded. So rewinding. Rewinding means already in our body is completely come to the normal position. This is all our unwinding position. Okay. So in this part started. This is the starting point. Active sites started from here. The RNA molecule synthesized in this part completely. So I will show you. So in the diagrammatic manner, it will be better for your understanding. Okay, so this structures. See, my dear students. Okay, so see the structure. This is a DNA structure. Imagine. So this is a. I draw here as a straight manner. Okay. So here, this is considered as a 5 dash and 3 dash n. Okay. Here, already in our board, 3 dash and 5 dash. In between these two, there is a molecule already in our board, the nitrogen bases, adenine, guanine, cytosine. Okay. All the molecules are united together with the help of the hydrogen bond, okay, and each side, both the sides, okay, it's on complementary to each other. Okay, the rho factor as well as the enzyme, the RNA polymerase recognize the place. For example, take an example, this place, imagine this is a recognizing place. Just imagine, okay, this is the place. Okay, here starting means here the adenine is there. Imagine here the adenine is there means what is that? A combination of other component. This is called as a RNA strand synthesis. This is the adenine always combined with the molecules of thymine, isn't it? In the case of RNA, the adenine molecules join with the uracil, isn't it? Here, the adenine join with the, sorry. Okay, see here. Okay, so see that. So this is called as DNA. This is called as RNA. This is a strand of RNA. How it will be formed? You know, starting point here, isn't it? Starting point is here. Here it contains which one? You know, so adenine molecule is there. Just imagine this is the adenine molecule. Okay, in this part there is a adenine molecule is there. So this is a DNA strand. Just imagine this is DNA strand. So with the help of RNA polymerase, recognition takes place. Starting point is here. This one is called as what is known? RNA strand. This is called as RNA. Okay, RNA strand is synthesized. RNA strand is synthesized, otherwise called as transcription. DNA to RNA synthesis takes place. The particular point is recognized already in our board with the help of RNA polymerase and rho factors. As a result, the initiation is also takes place. Initiation is called as what is this? Upstream movement in this part only. Okay, so the adenine molecule combined with the molecules of thymine nearest like this. So here, this point is the end point. Started from this only. Okay, I have to draw like this. For example, This is considered as a DNA. Okay, I have to start like this. So then only it will be correct. This is the starting point. So this is the starting point. Okay, so this is the starting point. After that, what happened? It continues developed. It is developed into the chain-like structure, single polypeptide chain. Here the starting point of adenine. So this adenine is in this first place only. This is the last one. This is the termination end, like that. It will be finished. Okay. So here, what happened? No, the structure is look like this, because it's scanning from starting from here to completely started scanning. After that, what happened? It is a starting point is here. So this is the first place. This is the first one. This is the first one. This is the 100 or whatever it may be. You can imagine. Okay, 100 nucleotide. So this is the starting point. So here I have told this is the first point. First point, adenine started here. And also this one is considered as a guanine, like that, it's a 100th place. There is a guanine is there. Okay. This entire structure is completely synthesized from the DNA molecule. That process called as transcription. This is how many nucleotides it may be? 17 base pair per minute. Already we studied. Okay. So that is the importance. Once again, repeat for you. This is the DNA molecules. The DNA molecules was recognized by the polymerase enzyme. Which polymerase is? RNA polymerase 1. Okay. It's a recognized position, particular position. That point is called as upstream. So the remaining things are after the transition process. This one is called as downstream. Okay. This is the starting point. The first one is called starting point. Okay. After the entire structure, this is the structure of the RNA molecule. Each one, it has a adenine, guanine, thymine, cytosine, like that. It comes like this. Okay. So this structure is produced from the DNA double stranded molecule. Okay. After the completion means already in our body. Initiation, elongation, and termination. Finished mean what happened? Automatically the RNA molecules comes down. RNA molecules completely synthesized. Okay. How many nucleotide means? 1 to 100. Okay. These molecules come down. Okay. Again, what happened? The unwounded. Unwounded means this part. Okay. This part completely rejoined together. As a result, form as a double stranded helical structures. Okay. This is the structure. This is a formation of RNA molecules from the DNA molecules. Okay. So don't forget, my dear. See that one, that things. See, so this is the DNA molecules. Okay. So unwinding means completely. That's why I have draw the DNA molecule in straight position. Okay. So you will get it. What is that? So then only the single one side only the entire molecules is synthesized. Opposite side there is no synthesis takes place. That is called as non-template stand. This one is called as template stand. Template stand. What is the mean? It produces the RNA sequences. Okay. So this is the diagram. See the transcriptions. They exhibit several features that are distinct from the DNA replications. There are many more molecules of RNA polymerase per cell than of DNA polymerase. RNA polymerase proceed at a rate much slower than DNA polymerase. This is very, very important one, my dear. Approximately 50 to 100 base per second for RNA versus near 1000 base per second for DNA. This is very, very important. RNA polymerase is very slow, slower than that of the DNA polymerase. DNA polymerase, it's up to 1000 base per second. This is 100. So 1:10 ratio, like that we can calculate, or 1:20 ratio. This one 50,000, 100,000, whatever it may be. This is 50 to 100. You just imagine this is a RNA polymerase. This is the DNA polymerase. Okay. The RNA polymerase lack of proofreading activity. The RNA polymerase, they do not have the proofreading activities of the DNA molecule because it's occurring only in the DNA strand only. Okay. It is not occur in the RNA. So that's why the RNA polymerase lack of proofreading activities. But at the same time, DNA polymerase, it has a proofreading activities. Okay. See that one, activities. Therefore, the error rate of transcription higher. than that of the chromosomal DNA replications. Okay, so it is also possible. What is that? Error rate of transcriptions. Sometimes it may be some other molecules, they may be joined together. Okay, the chromosomal DNA replications. When the RNA polymerase is activated upon, that time lack of proofreading activities, because RNA polymerase, they do not have the RNA proofreading activities. Therefore, the error rate of transcription is higher than that of the chromosomal DNA replication. Okay, so that unlike DNA polymerase, RNA polymerase does not require a primer to initiate the synthesis. Unlike DNA polymerases and RNA polymerase does not require a primer to initiate the synthesis. Only a very small portion of the genome is transcribed or copied into the RNA, whereas the entire genome must be copied during the DNA replication. Already you know about. Here what they are telling, you know, when the RNA production takes place from the DNA molecules, only the small portion of the genome is essential for the transcription of RNA. Take an example of DNA replication means, you know about the DNA replication is nothing but the entire DNA molecules completely copied, isn't it? But in this category, any small portion, that is a promoter only is enough. There is also promoter there. They also need a promoter, primer region, RNA primer is very, very essential. But entire sequence is not produced, isn't it? Entire sequence is not produced in the RNA, but DNA replication, the exact copy of the DNA molecules produced. Okay, so that is the difference between RNA and DNA. RNA means transcription, DNA means replications. At the same time, ribonucleotide are used in the RNA synthesis rather than deoxyribonucleotide. Ribonucleotide means ribose RNA synthesis. What is that? Ribonucleotides are used in the RNA synthesis rather than deoxyribonucleotides. So this is also another point. So go to the next one. The RNA polymerase require, in the addition to the DNA template, all 4 ribonucleosides, 5 triphosphates. Adenosine triphosphate, guanosine triphosphate, uridine triphosphate, and the cytosine triphosphate. These are the 4 triphosphate is also very, very essential for the RNA polymerase, for the template, all the nucleotides. It is nothing but the energy requiring process. Suppose the RNA polymerase is activate upon the DNA molecule, it needs some energy. Energy may be in the form of ATP, GTP, UTP, and CTP. ATP, GTP, UTP, and CTP. As precursor of the nucleotide unit of RNA as well as magnesium, the protein also find one zinc. So these two also very, very important. That's why it is called as, this is enzyme, this is the non-enzyme part, otherwise called as cofactor. So the both are united together, form as a coenzyme. Okay, so this is very, very important, my dear. This is a protein molecule and the energy requiring molecule, and also the cofactor is also important. So both are united together, form as a, what is that? Then we'll pass on to the next one, is called mechanism of transcription. What are the mechanisms? Already we studied mechanism of polymerase, already we studied. How it will be synthesized RNA polymerase, what is the main activities? That things already we discussed. Here, mechanism of transcription, how the RNA molecules are synthesized. What are the process? They are basically divided into 3 major steps. What is that? 3 major steps. One is called as initiation, elongation, and termination. So any one of the reaction, they need some promoter, initial step. And second one is the elongation. The process is continually going on until the requirement is fulfilled. And for after that, the termination. Okay, take an example of translation process also same. Initiation, elongation, termination. Starting codon is already know about AUG. That is denoted as methionine. When it will be finished, you know, complete 1500 or 10,000 or whatever it may be, complete protein molecule synthesized as top codon, when it comes last, that time what happened? Automatically the protein synthesis will be stopped. Okay, so that is the process. Here also the same process also occurs in the RNA synthesis transcription process. See that? So this is called as initiation process. This is the DNA helical structure. This is a rho factor starting from this. This is called as bubbles. Okay. With the help of the, which one, RNA polymerase enzyme with the sigma factors. It's also bind in the specific site, recognized by the specific site. Okay. That time automatically what happened, unbind the DNA molecules. Unbind. So during the unbinding DNA molecules, this is the RNA molecules produced. This is the RNA molecules produced. Okay. After the RNA molecules production completion, that process is called as, that means RNA molecules production takes place, that process is called as elongation of the polypeptide chain. Elongation. This is the initiation, means starting point. This one. And after that, what happened, the elongation, the entire molecule will be synthesized. Okay. After that, what happened now? There is a termination is there, the stop codon. Here the starting codon, here the stop codon. In between, there is elongation process. So as a result, what happened, see there, the double standard DNA molecule produce single standard RNA molecule. The RNA polymerase will be comes out. Again, it will be enter into the first step, initial stage, initiation, elongation, termination. The RNA molecule synthesized, it comes out. Again, the enzymes also comes out. It's also enter into the another substrate. Likewise, it will be continued. Okay. So this is the DNA molecules. The RNA polymerase find out the initiation or place. After that, it can move, completely read the molecules. This is the recognition. Okay. So recognize the molecules. That time what happened? It can move. During that movement, what happened? There is a bubble-like structures also formation takes place. After the end, what happened now? After the end, what happened? The complete RNA molecule synthesis takes place. The RNA comes out, and also the enzymes also comes out. Again, it enter into the another. The RNA molecules enter into the cytoplasm. Okay, see the initiation. Initiation involves the recognition of a promoter by RNA polymerase. Already I told you. The promoter region contains important sequences that are required for RNA polymerases to bind. These sequences are similar in both prokaryote and eukaryote genes, but the locations are different. That's why I told you prokaryote and eukaryote, it may be depends upon their activities. Okay, because the RNA polymerase, the prokaryote and the eukaryote, it can bind different locations. Different locations will be different. Okay, and bacterial promoters are approximately 40 nucleotide in length. Bacterial promoters, see that this is also very, very important. Bacteria means prokaryotic transcription takes place, means the initial point, the initial nucleotide. How many nucleotide may be present in the promoter region? Means 40. Take a note. 40 nucleotide in length in bacteria or prokaryote. They generally contain two short and conserved DNA sequences that are involved in the regulation of the transcriptions. One of these is found at the -10 position. Then 10 base pair upstream of the transcription start side. And as the sequence, see here, TAT, called the Bripnov box. Don't forget, my dear student, Bripnov box, the sequence is TAT-AT. This is called as, what is that? Bripnov box. Okay. The other is found at the -35. Here the -10, here the -35 position. And as the sequence, see that TTG, ACA. TTG, ACA. Okay. The sigma subunit recognizes the -35 sequences and cause the polymerase complex, leaving a 5 subunit core enzymes. Where there is a location means, what, but these are the 2 points is that this one is started from the -10. -10 is called as, what is that? Bripnov box. That Bripnov box contains the sequences is called as TAT-AT. TAT-AAT. Okay. The -35 position is the another sequences is that is called as TTG ACA. Okay. In this place only, what happened? The polymerase complex. What is that? Core enzyme as well as a rho factor. What is that? leaving a 5 subunit core enzyme in this position only. Okay, see that one. See, so this is called as DNA sequences. This is the promoter region, starting from the promoter region. This one is called as what is the -10 box. -10 box. See that TAT at. What is that? Consensus. -35 box. That means hereafter. This is the first place. +1. In this portion, a transcript region. In this portion, minus. Okay, this one is started from -1, -2, -3 up to 10, up to 35. Likewise, 100. You can imagine -100 like that. Okay, this particular blue color region, that is the -35 box only, it contains TTG ACA. So what about here? TAT and AT already told you. In between, how much molecules present in it? 10. So 17 plus 1 base pair. This is the main things. Already we discussed. Optimal interbase distances maximum 17 plus minus 1 base pair. Okay. This is very, very important, my dear students. This is the promoter region. This portion is completely called as what is that? Promoter region. This is going to transcript regions. Okay. So this is important things. See that? So 35 RNA polymerase. This is the 10. +1 started. ABC coding region. Downstream. This one is called upstreams. Okay. TA box is -10 and -35 regions. Already we saw it in the previous slide is clearly, isn't it? See that. The DNA unbound at the -10 sequence, the unwound region is known as the transcription bubble. Unwound is called as transcription bubble. Already we saw it. The RNA polymerase now begins synthesis RNA at the appropriate position on the gene. So this is the point. So where it started, see this position. So this is a place already know about starting point. So this bulb-like structure, bubbles. This is called as bubbles. Okay. A starting point here. Okay. So don't forget, my dears. It can move on it. As a result, what happened now? There is RNA molecule synthesis in the right to left. See that one. And second process is called as elongation. This is the starting point. And elongation, after the RNA polymerase attached to the promoter, the polymerase causes unwinding of about 2 strands of the DNA helix and separation of the unwound portion of the 2 strands. That's why it's straight positions. Then the polymerase move along the DNA strand temporarily, unwinding and separating the 2 DNA strand at each stage of its movement. Each and every movement, what happened? No, take an examples of just a double helical structure of the DNA molecule. When it binds with the RNA polymerase with the promoter, what happened? No, the DNA molecules, the helical structure will be unwinding. After the unwinding, the starting position is started. What happened? No, automatically it move just like a zipper. So it can move on to the DNA molecule in one side, means automatically the DNA molecule unwinding also started. Okay, unwinding and separating the 2 strands at each stage of the movement. Each and every movement of the enzyme molecule on it, what happened? Automatically the DNA molecules also, what happened? Move. As it move along, it's add at each stage. At a new activity, RNA nucleotide to end of the newly formed RNA chain. As synthesis continue and transient RNA DNA hybrid duplex is formed, but the RNA quickly dissociate from the DNA so that only the few RNA and DNA base pair are present at the given time. Within short period, what happened? The RNA molecule move upon it. As a result, this quickly dissociate from the DNA. Which one? RNA dissociate from the DNA molecule. As a result, you will get the RNA chain. Okay. A particular time, a given time means what is that? It may be 10 or 5 or 15 like that. The last one is called as termination. When the RNA polymerase reaches the end of the DNA gene, it encounters a new sequences of the DNA. What is the meaning of termination? That means what happened? Which codon it comes out here? Stopping codon, stop codon. DNA nucleotide called the chains termination sequences. The row termination depends on the action of the terminator protein called row factor. This is also another one. Row factor is also present. This causes the polymerase and newly formed RNA chain to break away from the DNA strand. When the row factor comes lost, means what happened? Completion of the transcription process. As a result, what happened? The DNA molecules, the RNA molecules comes from the DNA molecules, are dissociated from the molecules. At the same time, simultaneous polymerase enzymes also comes out. Already we saw it in. So then the polymerase can be used again and again for the still more new RNA chain. Already I told you, again and again it will be repeated. Okay, so the transcription is occurs with the 3 steps. So one is called as, what is that? Initiation, elongation, and termination. Initiation, how it will be, and elongation, how it will be, and also termination, how it will be. So termination process also done by the row factors. After the row factors, it comes out automatically. The polymerase enzymes also, what happen? Detach from the DNA molecule. It will also enter into the new process. Okay, so again and again they produce the new RNA chains. Okay, see, this is the complete entire structures. DNA strand is the, when it activated upon it, what happened? This is the bubble structure, unwounded structures. It can move like this until the end. So that time what happened? The single polypeptide chains also takes place. See that? So this one. So completely what happened? With the help of the RNA polymerase also removed. Row factors also comes out. Finally, this is the structure of DNA. The below there is a single standard RNA molecule formation takes place. Once again, this is a transcription unit. This is a transcription. What we are going to transcribe it. NEET. This is the starting region. That is called as promoter. When it RNA polymerase joined together, so this is the place, isn't it? That time what happened now? The unwounded DNA molecules. So the bubble formation takes place. After that, NTP, the RNA polymerase, the RNA synthesis takes place in this part. After the joining of the starting point, which one? Core enzyme. So it can move. See that the red lines started here. So this is the RNA molecule synthesis takes place at the initial state as template stand. The single standard RNA molecule is enough for this one. The entire RNA molecule synthesis. Okay. Replication process already in our report. Replication process, entire molecules, they are synthesis, and opposite side there is a leading strand, and also another one is called as lagging strand. Here there is no lagging strand, only leading strand only present in it. See that one? So here starting from the 3' to 5' end, it can move. It can be move on to the 5' to 3', but RNA synthesis takes place in the 3' to 5'. Final, what happened? These two are separated. That means DNA molecule separated and RNA polymerase also separated. After that, the row factor. Also, again, it will enter into the new synthesis of RNA molecules. So this is the process. Okay. See that one? Initiation, elongation, termination. This is the promoter region bind with the specific point downstream. It can move. During that time, automatically the RNA synthesis also takes place. When the row factors end to the last ones, okay, that is terminations. What happened? Automatically the RNA polymerase, RNA molecules also synthesized, detached from the DNA molecules. The row factors also comes out. Again, it bind with the, so this is the process, the initiation, elongation, termination. So we'll finish it up to this level only, my dear students. The importance of transcription and reverse transcription, termination, that things also we will see the next classes, my dear students. Okay, today what we have discussed already in our lab, what is meant by RNA polymer transcription. What is meant by transcription? DNA to RNA synthesis is called as transcription. With the help of the enzyme is called as RNA polymerases. Before the RNA polymerases, we must know what is meant by RNA polymerases, what are the types of RNA polymerases present in it. Okay, how they are classified. The RNA polymerases consist of the core enzyme and sigma factor already in our lab. The core enzyme is consist of the 5 peptide chain. Sigma factor is also a promoter region recognition factors on DNA molecules. After that, what happened? What is by holoenzyme? Already we know holoenzyme is nothing but the upper enzyme and the cofactor. Upper enzyme is nothing but the protein part, and cofactor is nothing but the what is that non-protein part. The both are collectively called as holoenzyme. Okay. What is the example? DNA polymerase 3 and RNA polymerases. Okay. So what will be the structure? Okay. These are the subunit present in it. So when the 5 subunits united together, that is called as RNA polymerase. Holoenzyme is the cofactor, and apoenzyme is also. So these are the things already we discussed. So next one is RNA polymerases. There is a structure of the RNA polymerases. See that 5. Okay. So, and what are the activities is that? One is called as initiation and catalytic activity and DNA binding. These are the 3 factors is also. 3 subunits activate upon the DNA molecules. Already you know about. So already eukaryote and prokaryote, what are the differences between these 2 polymerases enzymes? And then what happened inside DNA section at a rate of the 103 base pairs, less than 103. Okay. Until they recognize a certain specific region DNA in which bind with higher affinity. That is very, very important. Then only the transcription process occurs. Otherwise, it will not. Okay. So these are the type of RNA polymerases, RNA polymerase 1, 2, and 3. And then different classes also given here. These are the 3 things is there. And then, so how it will be? RNA polymerase 1, rRNA, RNA polymerase 2, mRNA, RNA polymerase 3, tRNA and rRNA, and also snRNAs. Okay. And RNA polymerase, what are the things? It has a subunit also there. This is also very, very important things. That things also already discussed. Mechanism of RNA polymerase already know a little bit. Upstream sequence, what is by downstream movements. Okay. So how it will be? Transcription takes place in the E. coli, particularly prokaryote. How many base pairs is very, very important. How it will be started from the left to right. Okay. So as a result, what happened okay, the bubbles formation also takes place. That things also initial stage, and with the help of the RNA polymerases, they can move on to the DNA molecules. It can synthesize a single standard RNA molecule. After the ending, row factors end with the termination end. What happened? Automatically comes out, and also started from the new one. It also involved in the new synthesis of RNA molecule. It is also separated from the DNA molecules. Already you know about that things also already we discussed. Okay. So how many base pair? That things also already we discussed, isn't it? That's all about today, my class, my dear students. We will see the remaining things on the next classes. Okay. Thank you so much, my dear students. Bye. Take care. Good night.
Thank you, sir.
Thank you. Thank you, ma'am.
Thank you, sir.
Thank you.